Welcome to Lecture 9 of CS 182. Today we're going to talk about how we can understand what convolutional neural networks are doing and how to actually use them to generate pictures. Before we get into the details of convolutional networks, let's briefly talk about some experiments, some very famous experiments in neuroscience done by Torsten Wiesel and David Hubel to study how real mammalian brains process visual images. And we'll see a lot of parallels to this when we discuss convnets. So Hubel and Wiesel are best known for their studies of visual processing in the mammalian brain. And their most famous experiment involved showing a cat different visual stimuli and recording the activations of neurons in the cat's brain. And what they found is that individual nerve cells in the cat's brain were responsible for recognizing different oriented edges. So this is a diagram of the experiment that they conducted. The cat was shown different oriented edges on the screen, and from this they were able to reconstruct how different neurons in the cat's brain were responding to different oriented edges. And that makes a lot of sense because objects in the visual world are delineated by edges, so if you can do a good job of detecting oriented edges at different orientations and in different places, then you'll have good visual processing. And in fact, these days, these experiments have been replicated extensively, and we know that early visual processing in the mammalian brain tends to recognize oriented edges, and the receptive fields of those neurons, basically the patterns that they most actively respond to, look kind of like this. They're kind of these little edges where one side is light and the other side is dark, and that tells you that there is an edge there. So we could similarly ask, well, what is it that convolutional neural networks see? Do they also see things in a way that's similar to how the brain sees them? And in fact, the early layers in these convolutional neural networks tend to actually greatly resemble those oriented edges that we saw in the Hubel and Wiesel experiments. But of course, then we could ask for our comment, well, that's what's happening in the early layers. What's happening in the later layers? Why are we interested in this question? Well, it can allow us to interpret what neural nets actually pay attention to, whether they're recognizing images, in a sense, for the right reasons. This can help us understand when our convnets will work and when they might not work. And it allows us to compare different models and architectures. Surprisingly, very similar concepts can also help us to manipulate images based on convolutional neural network responses to exaggerate certain features of those images. For instance, you could ask a convnet whether it sees different kinds of animals in the painting of the Mona Lisa and then exaggerate those features, or you could even ask it to hallucinate an image that would result if the Mona Lisa had been painted by a different artist with a different visual style. And we'll see how the same techniques that we use to analyze the responses in convolutional networks can actually be used to modify images in these creative and artistic ways. But first, let's talk about visualizing the responses in convolutional neural network filters. First, we need to ask, well, what is it that we should be visualizing? And we kind of have two options. One option is to visualize the filter itself. So let's not worry about images that activate or don't activate convnets. Let's just actually look at the filter itself and see if that can give us some intuition for what it's picking up on. And this is relatively straightforward to do for the first convolutional layer, because for the first convolutional layer, and this picture I showed, it was actually not from the mammalian brain, it was actually from a convnet, we can pretty easily imagine what kinds of visual features these filters will pick up on. So here's how we can think about it. These are pictures of a bunch of different filters, and we can say, well, what image feature does each filter correspond to? Well, take our image, like this dog, and then take one of these filters and imagine overlaying it on a patch of the image. And now ask yourself, if you take the dot product, if you basically multiply that filter with the image pixels and then add them all up, will that dot product be a large positive number or a large negative number? Or will it be kind of close to zero? So if it's a large positive number or a large negative number, that means that that filter responds to that patch very strongly. But if it's a small number, if it's close to zero, then it doesn't respond to that patch. So here you can see that the edge of the dog's ear has one color on one side and a different color on another side. So one of these oriented edge filters will probably react pretty strongly to that. In this case, actually, since the background is lighter than the dog, and the filter is going the other way, it'll actually have a large negative response. But you could imagine that maybe there's the negative of this filter somewhere in the network, and that one might have a large positive response. On the other hand, if you were to overlay this filter on the other side, where the edges are not lined up, so here the edge is going from the bottom left to the bottom right, but the dog's ear is going from the top left to the bottom right. Now here, because they're not aligned, the dot product between these will not actually be large, and therefore the filter will not fire at this location. So that's kind of how you can get some intuition for what the filter really represents. Unfortunately, this really only works for first layer filters, because the second layer filters are not expressed in terms of image pixels, they're expressed in terms of the activations of the first layer filters, and those are not nearly as interpretable to us. So another option that we have available to us is to try to visualize stimuli that activate a particular neuron. Now, what is a neuron in a convnet? Well, a neuron is a feature of a particular filter. And typically, when we're interested in the question of which visual stimuli activate which neuron, we're really asking that question over all image positions. So we could ask what activates feature number 37 at coordinate 2217 in layer 5. But usually we don't care so much about the coordinates, so the question we're more concerned with is what activates feature 27 in layer 5 overall at all locations on average. So we have our convnet, we can pull out some middle layer, and we can ask for some filter in that middle layer, what image patch will make the output of this filter large? Now, I apologize, the text is a little cut off in the video, but it says what image patch makes the output of this filter large. And we could actually try to generate image patches for which a particular filter in a particular layer outputs large numbers. And then we could look at what that patch looks like. And we'll find some filters that are receptive to faces, some filters that are receptive to dogs, and some filters that are receptive to more abstract things like rounded shapes. This manner of probing our convolutional network is conceptually actually very similar to the Hubel and Wiesel experiments that I mentioned earlier. Just like they placed an electrode in the cat's brain to measure which visual stimuli maximally activate which neuron, here we're placing a probe into our convnet to try to understand which visual stimuli maximally activate that filter. So let's try to do this. Visualizing the filters, that's pretty straightforward. You just directly print out the numbers in the filters. So if you have, let's say, 64 filters in the first layer of VGG, you can just lay them out in a grid with 64 cells, each one corresponding to a different filter. And you can kind of guess that some of these are looking at edges, some of these are looking at different frequencies, and some of them are looking at color patterns, and that's pretty much what you'd expect. In fact, it's a little bit striking maybe at first that the kinds of filters that we see in the first layers of most convolutional networks look extremely similar to the kinds of receptive fields that Hubel and Wiesel observed in the mammalian brain. And that's actually not an accident. In fact, while initially it was somewhat striking to computer vision researchers to see that convolutional networks acquire these kinds of filters, these days we actually know that almost any reasonable learning algorithm, when applied to real image patches, will actually discover these kinds of oriented edge filters, including k-means, independent component analysis, sparse coding, and many others. And the reason for this is not because all these algorithms work the same way that the brain works. It's actually because these are the dominant features in natural images. Natural images are not just random arrangements of pixels. They are presenting objects and scenes to us which are primarily composed of edges. So it makes sense that these networks would learn edges as their first layer filters very, very consistently. But again, we unfortunately can't visualize higher layers in a way that makes much sense. So for higher layers, we have to use option 2. So how do we visualize neuron responses? That's option 2. Well, one idea is to look for images that maximally excite specific units. So take a large collection of images, evaluate each of the filters in each of your layers for each of those images, and then sort them for each filter by how much they activate that filter. And then we can see the images that maximally excite each filter. So for some layer, pick some filter and says and ask what image patch makes the output of this filter large. So let me give you a little example of this. Let's say that I'm looking at filter 12 in layer 7 of VGG, just as an example. And let's say that the red box represents the receptive field to that filter. And the receptive field here covers about a quarter of the image. And I'm going to slide this receptive field over the image, and the number is going to represent the activation of that filter. So here it's 12.3, over here it's 37.4, over here it's 17.1, here it's 21.4, and here it's 42.1. All right, well, let me just replay that again. 12.3, 37.4, 17.1, 21.4, 42.1. So the question that I'd pose to you is, well, based on these numbers, what do you think this filter is actually paying attention to? What do you think it's looking for in the picture? And again, this patch is 42.1, this one is 21.4, this one is 17.1, this is 37.4, and this is 12.3. Right? So the thing that you might notice is that this filter tends to respond more strongly when the window is over the dog's eye, either its left eye or its right eye. So you might conclude from this that this filter is looking for eye-like features in images. And in fact, if we actually do this experiment, if we actually try to take a real network and look at the top regions for particular units, this is looking at units in the pool 5 layer. I believe this is for the VGG network, although it could be for AlexNet, I don't remember exactly. This is from the original RCNN paper. You can see that there are filters that are clearly receptive to particular, often semantically meaningful things. So the topmost filter seems to respond most strongly to people's faces and upper bodies. The second filter seems to mostly respond to dogs, although it also picks up on things with circles in them. So maybe it responds to dogs because it's really looking for like the 2 eyes and a nose, like 3 dark circles, and these boards with circular holes in them, as well as the American flag with the stars, looks very much like a surface with multiple circular features on it. So both dogs and fields of circles. The third row seems to pick up on roses and kind of arrangements of blobby red things, which appears to mostly capture roses and noodles. The fourth row appears to pick up on little bits of text, oftentimes the text 2006 for some reason. The fifth one, houses, and the sixth one, rounded, shiny shapes, including the shiny forehead of a bald man on the left, and the polished wooden tip of some piece of furniture in the sixth column from the left. So, not all of them are semantically super meaningful, like, lumping together shiny foreheads with polished wooden bedposts. Maybe it's not entirely reasonable, but visually they are similar, so maybe it makes sense that would be a mid-level feature in a ConvNet. All right. Now, we could do something a little different. Instead of just, this should say idea 2, instead of just finding existing images that already excite a particular filter in a ConvNet, we could instead try to figure out which particular pixels maximally influence the value at some unit. And that might give us a clearer picture of what exactly that unit is doing. By the way, when I say unit or neuron or filter, those are all synonymous with each other. So a neuron is kind of an older term to refer to particular numbers in the activations of a neural network. Those are more recently those are referred to as units, but in a ConvNet they're also referred to as filters, and those all mean the same thing. All right, so we want to find the pixels that maximally influence the value at some filter. And what does influence mean? Well, influence means that if you have some pixel, xij, and you have some unit, a at layer L, at position mn, and it's the filter p, so a, l, m, and p. If xij influences that unit, that means that a small change in xij should be reflected as a large change in the unit. And what quantifies this? What quantifies how much some value changes as another value changes. Well, that's exactly what a derivative does. So if we take some pixel xij and we take some activation and some filter, the partial derivative of the activation of that unit with respect to that pixel directly quantifies how much that pixel influences that unit. So if we care about AMNP at layer L with respect to Xij, then DAMNP over DXij is directly quantifying that influence. I should mention, by the way, that oftentimes we don't care about the activation of a filter at a particular position. We care more about the average activation of that filter at all positions, in which case we would sum over the position in the activation map and just look at the channel. But here, just for simplicity, I'm not doing that. All right, so how do we get this partial derivative? Well, it turns out that we can calculate these partial derivatives in the same exact way that we calculated derivatives with respect to parameters, by using backpropagation. And it's actually not all that difficult. So here is the pseudocode for backpropagation that we saw before. You have a forward pass where you calculate all the activations in your neural network, and then you have a backward pass where you initialize delta to be the derivative of your loss with respect to the last layer activations, and then for each layer from the end to the start, you compute the derivative with respect to the parameters at that layer, and we're going to not care about that because for now we don't care about parameters, and then you compute the delta for the preceding layer by just multiplying your old delta on the left by the Jacobian of that layer. And when you get to the very, very beginning, when you get to the very first layer, then when you update delta in this way, delta will actually end up being the derivative of your loss with respect to the original input, which is the image. So all we have to do in order to get backpropagation to quantify this influence is to initialize delta a little differently. Instead of putting a loss function on the last layer, we'll initialize delta at the layer that we want, at the layer L, to be the derivative of the unit we want with respect to the values at that layer. Now, this initialization has a very special structure. It's 0 everywhere except for the unit we want, MNP, because DAMNP over DAMNP is just 1. So you just initialize delta to be the same size as your activation map at layer L with a 0 everywhere except for the position you want, which you set to 1. So that's like your probe. Just like Hubel and Vazel had an electrode they put in the cat's brain, you're going to put a little probe into the convnet as just an activation map with a 0 everywhere except for a 1 in the place where you want to probe. If you want to probe the sum of some units, for example, if you want to probe the sum of the activations of a particular filter over all locations, then you would put a 1 at all locations for that filter, and then a 0 at all locations for all other filters. So the procedure set delta to be the same size as AL, and set it to be 1 for the unit you want to probe, and 0 for everybody else. Then run backprop from layer L all the way back to the image, and then read off the quantity you want, dA, MNP, dx, as just the delta you get at the last image, at the input image. So that delta, it'll be the same size as the image, and the numerical values in it will represent how much each pixel in the image affects the unit that you care about. So in terms of actually implementing this, it's actually very simple. You can use exactly the same backpropagation implementation that you would use for training your neural net, only now you'll train it to compute these derivatives of activations with respect to images. Okay, so let's see how this works in practice. Let's say that you have a network here that looks like AlexNet, and you feed in a picture of a cat. If you actually go and compute the derivative of some mid-layer activation with respect to every position in the input image, you'll get something that looks like this. Okay, it's not great. You can kind of tell maybe that it's more or less cat-shaped, although there's a lot of noise everywhere and you can't really tell what's going on. However, if you slightly modify backpropagation, you can actually get this picture. So I'll tell you what this modification is in a second, but in this modified picture, now it's actually a lot clearer what this unit is looking for. You can see that most of the grass in the background doesn't show up, and mostly this unit is emphasizing the cat's face. So it's looking a lot more at the cat's big eyes and nose and so on, and it's not paying as much attention to everything else. In fact, even for the eyes, the particular color channels themselves are actually meaningful. So you can see that in this gradient image, the eyes are actually blue, which means that if you were to take this cat and make its eyes even more blue, that would activate that unit even more. So it's a unit that is looking for big blue eyes. Okay, that's maybe kind of a sensible thing to do if you're trying to find cute kittens in photographs. So what's the modification? Well, the modification here is something called guided backpropagation, which is a trick introduced in this paper called Striving for Simplicity, the all-convolutional net. It's a little bit of a hack, and I don't have a really great explanation for why this is so important, but it appears to drastically improve the quality of images you get by analyzing these gradients. So the idea behind guided backpropagation is the following. A regular backprop is not very interpretable because lots of other units in the network will contribute both positive and negative gradients to a given unit. So this unit AM and P, it might be influenced positively by other units in preceding layers. It might also be inhibited. It might be influenced negatively by lots of other units. And these negative inhibitions, they tend to be very complicated, whereas the positive contributions tend to be a little simpler. It's not entirely obvious why that's true, but that just seems to be the case. So maybe if we just keep only the positive gradients, we'll avoid some of those complicated negative contributions and get a much cleaner signal. So what guided backpropagation does is it introduces a heuristic change every time you backpropagate through a ReLU. Ordinarily, when you backpropagate through a ReLU, if the activation before the ReLU is positive, then you just multiply your delta by 1, and if it's negative, which means the ReLU killed that activation, you multiply by 0. Here, what you do in guided backpropagation is that you also zero out the gradient if the incoming gradient is negative. So if the delta has a negative value in it, that negative value becomes 0 when it's passed backward through a ReLU. That shouldn't actually happen because in regular backpropagation, the Jacobian of the ReLU is 1 if the activation going into the ReLU is positive. So even if the gradient coming back is negative, that gradient should be backpropagated through the ReLU. And this method just says, no, don't do that, zero it out. It's almost like putting a ReLU on the backward pass as well as the forward pass. So why does this work? It's not entirely obvious. The intuition is that it kills off these negative gradients, these inhibitory gradients, and the inhibitory gradients tend to be much more complicated than the gradients that support a particular unit. So by keeping only the positive gradients, only the gradients that make the unit fire more, and removing the ones that make it fire less, it seems like we're able to recover much more interpretable impressions of what a particular unit is looking for. But it's a little bit of a heuristic. Okay, so if the way that guided backpropagation works is still not entirely clear, maybe this picture from the paper will help illustrate it. So here, FLI is the activation of the unit before the ReLU. R, L plus 1I, is the delta after that layer. And what you can see is that regular backpropagation just sets the derivative at the layer to be the derivative at the next layer multiplied by an indicator for whether or not the unit is positive. So that's because the derivative of the ReLU is 1 if it's positive and 0 if it's negative. And then guided backpropagation, which is the thing in the bottom row, also zeroes it out in addition if the derivative itself is negative. So only the positive derivatives get backpropagated. Okay, a little bit of a heuristic, but that's how it works. So here's what the authors of this paper came up with as some visualizations of different units in layer conv 6 and layer conv 9. I believe this is for VGG, but I'm not completely sure. So the way that they generated these visualizations is they first found the patches that maximally activate different units, and then they used this gradient method to figure out which particular pixels in those patches are responsible for that activation. So you can see that for conv 6, these are some lower level features. The patches that activate them seem to be, let's say in the first row, kind of circles that predominantly represent dogs' noses, but not always. And the particular pixels are actually the noses themselves. In conv 9, this is higher up in the network, so the receptive fields are larger, the features are more complicated. And here you can start seeing some more interesting patterns. You can see a clear preference for people's faces in the second row, a clear preference for circles in the third row. And you also notice that in each image, some things are actually removed. Like, for example, in the bottom image, in the bottom row, this is a unit that appears to have a preference also for circular things. So for the cowboy on the left, for the man wearing the cowboy hat is very heavily emphasized, whereas the shirt is comparatively less emphasized, especially towards the bottom of the image. So this is really picking up on particular properties of these patches, not just the entire patches themselves.